our first speaker will be Adi Mukund, and it's going to be my great pleasure to introduce her. She's a MD PhD student at Stanford University with Bingdu, where she studies transcriptional activators as well as repressors with high throughput screens. And she actually got her bachelor degree in computer science as well as mathematics from Duke University. So it's quite a switch. And she has won several awards as well as taught multiple classes, and which was like really impressive. One thing I really want to highlight from her PhD is her involvement in promoting diversity as well as equity in Stanford University by involving in multiple advisory groups serving as the broad members or organizers. So it's really a lot of effort in promoting diversity and equity. So I will hand the floor to her to talk about her thesis work on high throughput screenings on transcriptional activators and repressors. So Eddie, you can go ahead and share your slides.
Yeah, thank you. That was an incredibly generous introduction. Yeah, I am delighted to share with you all kind of the bulk of my PhD work, which is on characterizing these combinations of transcriptional effector domains, both activators and repressors. The impetus for this talk was kind of thinking about how our understanding of transcription factors was. And a few years ago, we often thought about them, at least in the Bintu lab, with a picture that looked kind of like this, where you have a promoter or an enhancer and a DNA binding domain that would localize the transcription factor to your targets, and then an effector domain that kind of did a lot of the work once a transcription factor was bound. So recruiting cofactors, modifying chromatin, doing a bunch of other things. And over time, it became clear that this picture wasn't quite right. I mean, I'm sure it's not right for a number of reasons, but one of the big reasons is because oftentimes you have a second effector domain present on these transcription factors. And so DNA binding domains are known to kind of exist in these combinatorial ways. But as the evidence mounted that effector domains also kind of had this combinatorial interaction, we became curious kind of how that worked, what the rules of those interactions were, and how effector domains behave when you put them together that was different from how they behaved when they were on their own. The way that we kind of operationalized thinking about this when looking at kind of pairs of activators or pairs of repressors or activators with repressors was to think about how they behaved when you paired them up versus when they were paired with kind of non-functional or control domains. And the reason we operationalized that this way is because it's not immediately obvious that a transcription factor effector domain that's paired with another effector domain would behave the same as when it's on its own, even if its partner isn't doing anything. And so the way we tried to organize this was to map the strength of a combination of effector domains versus the sum of their individual strengths. And so if your strength of the combination is much higher than the sum of the individual effector domains measured strengths, we think of that as kind of being synergy, where you put them together, they work better than you would just imagine based on how they work on their own. You could also imagine kind of an additive mode of interaction or of the pair working together, where the strength of the combination is just like a straightforward sum of the individual strengths, or kind of antagonistic behavior, where each of the effector domains might work really well on their own, but if you put them together, all of a sudden the molecule isn't nearly as effective at activating or repressing gene expression. So we wanted to kind of build a plot like this that comprised lots of pairs of effector domains and see if we couldn't get at rules of how effector domains work when you pair them up. So our approach to this is kind of heavily based on some of the previous high throughput work that our lab has done, pioneered in part by Josh Tycho. And so we took a panel of about 100 or so effector domains and we put them in one of two positions, either kind of in front or behind the other one. And we recruited them in a pooled high throughput format to a synthetic reporter locus that we installed in K562 cells. This reporter expresses not just a fluorophore, but also a surface epitope that you can use for straightforward magnetic separation between on and off populations. And by sequencing which domains are in the on or the off population after some period of recruitment with doxycycline, you can get a sense of if an effector domain is turning genes on relative to control domains that don't do anything, or turning genes off relative to those, and so on. You can measure activation and repression by more or less just swapping the promoter. If you put a promoter in front of the reporter that's relatively weak, like the MinCMD promoter, then you can very easily measure activation. And you can also measure repression to put a very strong promoter in front, like the human PEF promoter. And so we did this for about 8,400 domain combinations. We computed what are called enrichment scores for activation and repression. And we were pretty happy to note that these high throughput measurements were fairly reproducible. You can see kind of in gray on both of these plots a blob that corresponds to pairs of negative control domains that we didn't expect to do anything. And then if you kind of draw a threshold a little bit away from the mean of those control domains, you can see a fairly large set of domains that have varying strengths of activation or of repression. And so we were excited by this in part because we hadn't really tested these combinatorial effect domain sets before, and also because I think at the time we were having potentially a little bit of trouble, but we fixed a lot of the shenanigans that were happening. And so we had these kind of high throughput measurements that were nice, reproducible for measuring activation and repression. And so the first thing we really wanted to do was get a sense of what each of the effect domains that we had in this panel were doing kind of just with negative control domains. And so we kind of built a plot that looked something like this, where if you have low activation and low repression with negative control domains, then your effector doesn't really do anything, and we'd call those a non-hit. You can also get effector domains that would only activate or only repress. But you could also imagine effector domains that in combination with non-functional negative controls would be able to both activate and repress different promoters. And so we'd call these dual functional effectors, although I think they're often sometimes also described as bifunctional effector domains. And so if you kind of build this plot of looking at how each of our effector domains works either as an activator or a repressor, a non-functional domain, or a dual functional domain, you get a plot that looks something like this, where you have kind of this nice long line of repressors on the left, you get some activators on the top, and intriguingly, especially for us, a lot of our strongest activating domains were dual functional domains. And the classic example of this that we saw was the FOXO3 transcriptional activating domain, which also worked as a fairly reasonable, albeit kind of weak repressor in our hands as well. But we were excited, I think cautiously optimistic once we put this panel of labels together to try to figure out, once we could understand the individual behavior of effector domains, how do they work now together? And so the first part of this question was to look at pairs of activator domains. And so this would be like CRTC2 with CRTC2 or with FOXO3 and so on. And when we looked at pairs of activating domains, we got something that looked a little bit like this, where you have pairs of control domains in gray, activator pairs in yellow, activator and dual pairs in orange, and dual pairs in red. And the thing that leapt off the page to us immediately was that these activator pairs do way better together than they do, or than you would expect them to do if you're just taking the sum of their individual strengths. And on the flip side of this, your dual pairs tend to do a lot worse than you would expect by just the sum of their individual strengths. Our first hypothesis here was this was just kind of an artifact and it was all just promoter saturation. So we decided to test a bunch of these in low throughput to verify that we weren't just making up conclusions. So the first thing we did was we tested this ANM2 Kibra pair, which individually didn't seem like they'd be particularly great, but together were really, really strong. And that is what we saw. We recruited them in low throughput to our reporter for a couple of days. We measured expression by flow cytometry. And ANM2 on its own doesn't activate very much. Kibra on its own activates a decent amount. You put them together and you get a ton of activation. They work really, really well. So it didn't seem like our synergy kind of conclusion about weak activators was entirely off base, but we were still quite skeptical of, for example, the MIB-A ZN473 pair, because each of those works as a really, really strong activator in our hands. And we were like, how on earth could these together not work very well? But actually, when we tested in low throughput, that is precisely what we saw. MIB-A on its own was a very strong activator. ZN473's kind of variant crab domain, very strong activator. You put them together and they don't work well at all. We don't think this is due to self-fitness effects, in part because each of these effector pairs also has an M-cherry fluorophore that we gate on, so it's not like the transgene is just being silenced. And the cells were dividing at totally normal rates. So while mechanistically, I think we're still not sure what the cause of this antagonism is, we're fairly confident that we are in fact seeing this, and it's not just an artifact of our system. So the overall takeaway, I think, from our examination of activator pairs is that the weak activator pairs really synergize quite well, but strong activator or dual functional effector pairs often will antagonize each other. The flip side of this question is to look at repressor domains. And here we see something that's a little bit different. You see a fairly nice line from the gray control domains along the kind of dashed y equals x line, and then a little tail of repressor pairs that goes off towards the left of the screen. And so while last time kind of that tailing off behavior reflected real antagonism, here we wanted to know, like, okay, is this antagonism or is it just, again, in this case, saturation, because you can't silence more than everything? And at least with repressors, the answer was it's saturation. ZNF10, CVX1, both independently and together totally turn off all gene expression when we measured them in low throughput to verify. And we're working to figure out if that linear behavior you see in the graph holds up, especially when looking at weak repressor domain pairs. But overall, the observation here was that repressor combinations generally work really well to silence the PEF promoter, and they don't appear to antagonize each other in the same way that we saw activator pairs do. This plot here was like a week of pain for me to generate, but it shows, I think, the main conclusion of our combinatorial work, which is that in our hands, repressors tend to overpower activators. Each row or column is a particular effector domain. The dual functional effectors they have both rows and columns for them. They're sorted first by kind of the type of domain they are, non-functional domain, activator, dual functional, repressor domain. And then within each of those categories, their strength on their own when activating or repressing a gene. And the first time I made this, I was kind of blown away by how the right-hand side was just all blue, like just everything. You have a couple of domains that can weaken certain strong repressors or kind of break them, but by and large, you put a strong repressor with something else and you're just getting repression out of there. Life gets a little more interesting when you look at pairs with dual functional domains and other types of domains, especially if you pair them with an activator domain. Oftentimes you lose that repressor function, which is kind of an interesting observation we had noted. And when you pair dual functional domains with each other, you mostly just get dual functional domains back again. I think, to our knowledge, this was the first time that someone had shown this in what was a relatively systematic way. I think there have been examples of repressors overpowering activators and so on before, but those tended to be individual examples. So we were really kind of excited and happy to be able to put that out into the world and see if it held up as other people, tried to poke at the conclusions. After we did all of this work, the question we asked ourselves was like, okay, we have all this information on how pairs of effector domains work together. What can you do with pairs of effector domains that you couldn't do with just a single effector domain on its own? And the way we tried to do this was we were like, what tool can we build with a pair of effector domains that we couldn't build with one effector domain on its own? And to us, the obvious way to go about thinking of this was to put something in between the effectors that you could cleave off kind of in a titratable way, such that you could go between having both effectors or just one effector. And to do this, we used these staple constructs from Michael Lin's lab, just, I think, up a floor in our building at Stanford. And the idea here is the staple domain is basically a protease with its cleavage site right next to it. So in the absence of a drug ASV, it cleaves itself off along with the ZNF10 crab domain, and you should get just a DNA binding domain with the dual functional FOXO3 TAD. But then you can stabilize that linker with ASV, and you would get FOXO3 with this little thing in the middle, and then the ZNF10 strong repressing crab domain. And our hope was, without ASV, the staple TF should look just like FOXO3, and with ASV, it should look just like crab. Luckily, this is pretty much what we saw when we recruited it to the minimum CMV promoter to measure activation. Without ASV, it activates very similar to how FOXO3 on its own activates. With ASV, there is no activation, just like crab. And we can play the same game for repression. And once again, we see that without ASV, the staple TF behaves very similar to the FOXO3 transcriptional effector domain on its own. And with ASV, it behaves very similar to crab. And so it was nice, the system was working as intended. So the follow-up then is to try a bunch of different doses and see, as you vary the levels of doxycycline for recruitment and ASV for keeping crab on or keeping it off, how does the behavior change? And so if you have kind of a reasonable dose of dox and increase the amount of ASV, you go from a profile that's mostly activation to no activation whatsoever. And if you have no ASV, you can slowly increase the doxycycline dose and get kind of increasing activation with more recruitment of FOXO3. And if you kind of plot all of these doses out on a little grid, you get something that looks like this, where you need fairly, non-zero doses of dox and very low ASV doses to get measurable activation. And anything more than almost no ASV will get you measurably strong repressors. And especially when you get to 0.1 or 1 micromolar of this drug, you get a lot of repression. And so the way we tried to figure out, like, okay, is this giving us something that we wouldn't get from an individual effector domain alone, was to look at a plane that's shaped like this. So each dot on this plane is going to be a particular time point of recruitment. So recruitment for 2 days, recruitment for 3 days, recruitment for 4 days. And the x-axis is going to be the fraction on from the min-CMV promoter. So how much activation do we see? And the y-axis is the fraction on from the PEF promoter, which is a proxy for how much repression we see. And what we see is that at the extremes of ASV levels, we get kind of lines either along the left-hand side for maximal ASV doses, which we think is similar to crab, and along the top curving down a little bit for no ASV, which is similar to FOXO3. But as you vary the dose, you get a lot of points floating around in the middle. And this was kind of our way of visually confirming that actually, when you have multiple effector domains, especially with this kind of interesting tunable cleavage site in the middle, you can get behavioral profiles that an individual effector won't give you. And so that is kind of where I'd want to wrap up. A bunch of people to thank in the Bintu lab, Lacra, of course, but also Sage, Cece, Connor, and Steph. Josh this project couldn't have happened without him. He was really helpful in getting some of the early parts set up. We have a bunch of collaborators in the Basic lab who I'm really indebted to. And then Anshul and Will are both also at Stanford and provided some really helpful input. And yeah, with that, I'm happy to take questions.
Thank you, Adi, for this great talk. Okay, so moving to Q&A. If you have a question, you can either raise your hand and ask your question, or I can ask your Q&A box. So Kaya Mattioli asked, very nice talk and cool work. Did I see at the beginning that you tested effectors in 2 orientations? For example, 1+2, then 2+1. Did that have any large effects?
Yeah, this is a great question. It was kind of one of the early things we looked at. And thankfully, I have a slide for it. So we actually observed relatively few cases of what you consider statistically significant orientation-dependent function. This is looking at domains kind of paired mostly with negative controls, just because those don't really do anything in it. So we thought it was a good test for if a domain's function really depended on orientation. And across kind of all of the different combinations of like activation or repression measurements across effector domain, we only really see 4 examples of domains whose effect is significantly changed based on its orientation just with negative control domains. And so for most of our analysis, we kind of excluded orientations that significantly broke the function of the domain because we didn't want things to just not work because of the order we put them in. This is, I think, kind of different than what our intuition was and what a lot of work has shown, which is that depending on your orientation in synthetic constructs or transcription factors, you can get very different effects. But at least in the context of this assay, we didn't see a ton of that problem show up.
Okay, so the second question is from David Arnosti. David asks, in simple additive or synergistic activation, those AD targeting based on machinery, or non-additive synergistic effector domains that bind to co-regulators such as HAT, etc., etc.
Sorry, I do not think I understood the question. Let me see.
Simple additive or synergistic activation, those AD targeting based on machinery, or non-additive or synergistic effector domains.
Like, is there a pattern to the effectors that
basically,
I think this is a good question. The answer, at least for us, was that strength explained virtually all of these differences, and it tracked a lot less with like what those effector domains did. We I think saw that especially for domains that we thought interacted with basal transcription machinery, there was like a fairly wide range of how strong those were at activating. And so their strength of activation was more predictive in our hands than their mechanism, which was interesting. I don't know if I would say in general that that's like a reasonable or like the best conclusion, but from what we did, that seemed to be what we saw.
I think Ben had also one question. Ben, do you know?
Yeah, very cool talk. So for a gene to get turned on, if it's repressed, does this say that, you know, we often think about a transcription factor becoming expressed and going to activate the gene, but in your system, when the repressor is the one that wins more often than not over the activation domain, does this mean that we need to think about how to prevent the repressor as much as turn on an activator?
Yeah, this is a good question. When I say the repressor wins, I guess I mean kind of two things at the same time by that. One is that if you recruit the activator repressor fusion to a weak promoter, you don't see activation. And also that if you recruit the repressor activator combination to a strong promoter, you actually see repression. This was a fairly consistent conclusion in our hands. I know it's not the first time. I think there's a particular transcription factor with like a C2HR and a crab domain that kind of antagonize each other in a similar way, and the repressive C2HR domain tends to win out. I mean, I guess the short answer to your question is yes, but we didn't do a ton of work to like figure out at endogenous genes how this works. We did do a couple of experiments for like if you recruit a repressor for a little while and then recruit the activator versus if you just recruit an activator. And those are in the preprint, and we find that it's very different depending on like the temporal ordering of those two. So I hope that gets a little bit towards answering your question.
Absolutely. Thanks again. Fantastic talk.
Thank you.
So there is one more question from Angelina Zion. I will unmute Angelina so she can ask a question. Angelina, you can ask your question.
Hi, can you hear me okay?
Yep.
Yeah.
Awesome. Hi, I'm a trainee. Great talk. Thank you so much. I was wondering if when you're talking about this, when you're talking about activation, what parameters exactly maybe I missed this, but what parameters exactly were you looking to measure that with? Did you look at all at, sort of a more kinetic characterization of, did this impact like the frequency of the burst or the, the amplitude of the burst peaks versus the duration of the burst, with anything like MS2 or something like that? And whether, if you did, were there any, were these parameters different in how these combinations affected them?
Yeah, I think this is actually a great question, and apologies if I didn't make it particularly clear enough. The way we ended up doing this was with kind of a synthetic reporter system, where you recruit these effector domain pairs to, I guess for activation, a weak promoter that drives a synthetic kind of surface protein, and we look at kind of presence or absence of that surface marker in the high throughput setting to measure gene activation. We were kind of hoping, or like, yeah, we were vaguely hoping after the fact to be able to think about things like burst size or frequency. But ultimately we found that just the kinetics of like degradation and production of our reporter didn't allow us to do that. We didn't end up doing any like MS2 stem loop related style experiments to see which of these affected, burst size versus burst frequency and so on. I know was some work recently, and I wish I remembered the author's name, looking at kind of like a kinetic landscape of transcription factors, and they found that some TFs predominantly affect one of those two parameters. We just didn't have the resolution in our system to be able to ask the same question.
Gotcha. That was Nick Mamra. I'm actually from his lab.
Oh yeah. No. Saw that and I was like, wow, this is a really great paper. We should see if we can do this in ours. And then no, we can't. We're very sad about it. But yeah.
Yeah, thank you so much.
Thank you.